We'll start with a short talk by Michael Robson. He's from the Max Planck Institute in Berlin, and he will talk about multi-omic single-cell profiling in the context of nuclear envelope release and gene activation in mouse during mouse embryogenesis. And we are very much looking forward to your presentation. Michael, I hope everything is set. I know you have presentation mode now. Yes, it's there. And yeah, the stage is yours.
Okay. Hello. Yeah, you can hear me. Okay. Yeah, so it's a great pleasure to be here and present our work, where we've been working with my PhD student Konrad Hutzig to look at how the nuclear envelope can regulate gene expression during development. Let me just put my pointer on. So Anna's already given a really nice introduction about 3D genome structure. And one of the biggest observations about how the genome is organized is that much of the genome attaches to the nuclear envelope in these repressive lamina-associated domains or LADs when it's inactive, whereas much of the active genome positions in the nuclear interior in non-LADs. Now, perhaps unsurprisingly, considering these two zones are associated with different activities, many genes need to actually transition between LADs and non-LADs and during development as they are regulated. And we can directly see this, for example, at the ZFP42 locus by super resolution fish microscopy, where you can see the ZFP region shown in green here is away from the red stained nuclear envelope when it's active in ES cells, but it moves close to the nuclear envelope when it's inactive in differentiated cells. However, the big question is, well, considering there's this relationship between gene position and gene expression, which feature is driving the other? And people have been limited to looking only in vitro settings, often using artificial experiments to test this relationship. So, for example, people have taken active loci that are located in the nuclear interior and forcibly dragged them to the nuclear envelope, and they see this drives gene repression. So in many cases, so in this case, gene position is controlling gene expression, but at the same time, people have also taken inactive loci and artificially activated them when they're at the nuclear envelope, and this repositions them to the nuclear interior. So it sort of remains fundamentally unclear whether gene position or expression is a cause and consequence of the other. And this is what Konrad and I wanted to look at for his PhD. We wanted to establish what the causal relationship is between LADs and transcription. And to do this, we wanted to take advantage of these new single cell approaches to simultaneously measure LADs and mRNA in a genome-wide manner in single cells, but do this in a process of development in vivo, where cells are exploring many different possible developmental trajectories. Sorry. And to do this, we paired up with Job's Kind's group at the Hubrecht Institute, who've developed a technology called single-cell DAM&T, and we've been working that simultaneously profiles LADs and mRNA together in single cells. And we've been working with Isabel, Sami, and Robin to apply this in vivo. So briefly, what single-cell DAM&T does is it first takes single cells and uses a modified cell-seq protocol to quantify mRNA, where we basically perform cell lysis, mRNA reverse transcription, and second strand synthesis. And then in the second step, we can progressively, use a technology called DAM-ID, whereby we take the bacterial DAM methylase and fuse it to a protein of interest. And then this will methylate nearby GATC sequences in a way which isn't normally present in mammalian cells. And by fusing this protein to a nuclear envelope protein like lamin B1, we can then progressively label LADs over time. So after doing your mRNA processing, you can then move to the genomic DNA, use a methylation-specific enzyme DPN1, which only recognizes methylated GATCs. And this will then cut our labeled LADs, and then we can tag these with an adapter through ligation, and this has a unique barcode. And then using unique barcodes in each of these steps, we can then generate Illumina sequencing libraries for single cell analysis. But the problem with this technology at the time was that it only functioned in vitro and was very leaky, which means we wouldn't be able to get the time resolution that we need for our in vivo experiments. So the first thing Konrad and I set about doing was generating a mouse system where we could generate embryos that harbored a tamoxifen-inducible CRE, and on a second allele, we would have a constitutively active promoter that would drive the expression of fluorescent reporter, M-cherry, with a strong transcriptional terminator that is flanked by LOXP sites. And then when we inject the pregnant mothers, this can allow the tamoxifen, the CRE, to suddenly excise this stop cassette and enable the direct expression of GFP, as well as the DAM-Lamina-1 fusion protein, which can then progressively label LADs over time. And in these embryos, we can then take any tissue that we're interested in and then generate a single cell suspension that we can subject to FACS. And we can then sort individual GFP positive cells into a 384-well plate, which we can then process into the single cell DAM&D pipeline. And basically through barcodes introduced to the mRNA and genomic DNA processing steps, we can then pool all the cells, amplify all the material together using in vitro transcription, and then generate Illumina libraries. So with this system, we had to decide on a developmental model that we were going to analyze. And in the end, we settled being from the Munlo slab on the developing limb. And this is a beautiful model of a highly spatially and temporally precise development system that is very well understood. So in the limb begins in E85 embryos as this homogeneous population of mesenchymal stem cells that is encased in a layer of ectodermal cells. And these mesenchymal cells progressively differentiate in a highly spatially and temporally precise manner to produce bone, cartilage, and connective tissue. But at the same time, we also have a second wave of incoming cells that migrate into the limb from a completely different progenitor source that will go on to produce muscle, blood vessel, and neuron. So this provides us with many different trajectories that we can analyze this relationship between LADs and transcription. And in addition to this, we can also analyze outgroup tissues like brain, lung, gastrointestinal tract, kidney, and heart for comparison. So we've now profiled all of these stages in over 8,600 cells. We actually have more now, but I'm only going to show you this preliminary data set. And we can first look at the transcriptomic part of the single cell DAM and T data and see how these, if we can identify distinct cell types. And indeed, we can see that the cells cluster in UMAP space according to their transcriptional identities based on the cell type, the tissues that they originate. So on the bottom here, we have the limb cells. On here, we have cells that come from the E85 embryo. And here, for example, we have cells from the brain. Now we can use clustering and marker genes to then identify specific cell types in this data set. And we can see this limb cluster down here is associated with the limb mesenchyme. And this is closely affiliated with cells from the E85 embryo, the mesenchyme where the limb, all these limb cells originate. We can also see we get these migrating skeletal muscle cells from the limb, as well as endothelial cells here. So we can identify biologically meaningful cell types just from the transcriptomic information alone. And we can even label cells according to the stage that they originate from in development. And we can see, for example, in the limb mesenchyme, that these limb mesenchymal cells here originate at E95, and then they radiate out progressively in E10.5 and E11, going into E13. So it means that we can recover the developmental trajectories that cells undergo using the transcriptomic data. So the big question is, well, considering we can detect transcriptomes, can we detect LADs in these different cell types? And the answer is yes. So here we can look at the LAD profiles for individual cells going in these individual rows, the entire of chromosome 1 in, for example, ES cells and endothelial cells. And what we can see is we can reproducibly get very nice LAD profiles in each of these single cells. And we can even get cell type specific differences between the LAD profiles. We can sort of quantify what degree of tissue specificity we have for these LAD, these nuclear envelope attachments by asking in how many cell types is any given region of the genome found at the nuclear envelope. And we can see there's a fraction of the genome which is never found at the nuclear envelope, and there's sort of a constitutive non-LAD, CI LAD. And we also have a fraction of the genome which is always found in the nuclear envelope in every cell type we've looked at. So these are constitutive LADs. But the rest of the genome we see is in this facultative category. And basically, because of the different sizes of these regions, we can see that these constitutive non-LADs account for 30% of the genome, constitutive LADs reflect about a third as well, and the remaining 36% are all tissue specific. So the single cell DAM and T method allows us to simultaneously profile LADs and transcription in these really complex tissues, and we can already see that LADs can be highly tissue specific. And now, just for time constraints, I'm just going to try and describe to you how this laminar association then reflects to gene expression by just looking at 2 examples to give you a flavor of what the data can show. So we can look at, for example, the activity of the GAP43 gene, which is active in a number of neuronal different cell types. Now we can ask, when it's active, what's its laminar association? And what we see is that when the gene is active, you have low DAM-ID signal in the cell types where it's active, but not in the cell types where it's inactive. So what this tells you is that laminar association being repressive is fundamentally coupled to a gene and being inactive, and there's a coupled relationship between the two. However, we see that it isn't true, surprisingly, for a number of loci. So for example, with SHOX2, which is a gene which is active in a number of neuronal cell types, as well as in multiple different cell types in the limb, we see a different picture. So for SHOX2, we can see that in the brain, you have it locally having low DAM-ID signal when it's highly active in these cells, and it seems to be coupled in this tissue. But when we look in the limb, we can see, although it's only active in a small subset of the overall limb mesenchyme, it's completely non-laminar associated in every cell from the limb mesenchyme. And it is even true, we can trace this back to the developmental trajectory of the limb. We can see that the detachment already begins at the earliest stage of limb specification and isn't present in the E85 embryo mesenchymal cells where the limb originates. So it seems that laminar association can be uncoupled, and this happens very early on in the specification of the limb. So sort of, I think that's just the flavor of the data that I think we can generate, where we can show that indeed there is highly dynamic and cell type specific nuclear envelope attachment in development, and that we can, and in situations this can be coupled fundamentally to transcription, but in many other cases, this cycle is broken between them, and genes can detach from the nuclear envelope independent from their activity. And our hypothesis currently is, in the case of a number of genes like SHOX2, is that this is happening, the nuclear envelope release is happening in progenitors far earlier than the gene needs to be activated as a way of priming the gene and its regulatory landscape for activity in later cell types. So with that, I'd just like to acknowledge people I think have acknowledged them along the way. I'd like to thank Conrad, who's the PhD student who's been doing a lot of this work, and all our collaborators from the KIND lab who've been working with us on this, and also our mouse facility at the MPI, because we wouldn't be able to do any of this work without them. With that, any questions are more than welcome.
Thank you very much for this highly interesting talk about LADs and LAD dynamics, raising the fundamental question, cause or consequence, as usually in biology. And as you can see, there is no black and white answer, apparently.
Complicated, yeah.
More complicated. So Anna wants to ask a question.
Just, one question is in terms of the silencing mechanism. So do you think that when SOX2 is at the periphery, it's being at the periphery that's repressing it? And equally, how is it repressed when it's not at the periphery?
Yeah, so I think what's clear is that SHOX2 is just completely inactive, even though it's been released from the nuclear envelope. So I think nuclear envelope attachment is not, or loss of nuclear envelope attachment isn't sufficient to cause activation, but I think it might be necessary for the gene to later become turned on. So we've been looking at, so SHOX2 is a polycomb gene, so it could be that it's being actively silenced until it needs to be turned on. But I guess also its regulatory landscape and enhancers are just not engaged in that in these earlier stages. So a really interesting question would be, if we force it to the nuclear envelope in the progenitors, can we prevent it turning on later? And then the real question is then, well, if transcription isn't releasing the gene, then what's causing the entire landscape to detach? And we have some ideas for this.
Okay, thank you very much. Are there questions from the audience in the chat? I don't see anything at the moment. There's Gonzalo.
Yes, I have a question. Yes, I'm just wondering if you have single cell ataxic data sets that you can overlay, because it will be interesting to see if the priming, if these are different levels of priming, one that is at the lamina and then chromatin accessibility and so forth.
Yes, so we're currently doing this single cell ATAC and transcriptomics, like in the joint assay from 10x, and we're going to do this at this transition point when these limb progenitor cells go from having the attachment to not having the attachment. But I think that's a beautiful question. I want to know what's driving the release. I guess it might be that the accessibility landscape is changing prior to everything else. But.
Thanks.